Okay, this PowerPoint video will go over the outline I gave you for test 2. So I'll go over each one of the parts. Hopefully, it'll help you out. But you're also going to have to, I'm not going to be super explicit about all the answers. You're going to have to check your notes and the videos to answer some of it. But, I'll give you an idea what to think about. It's sort of like the class. And it's like a review for the class. Can't cover everything in a short amount of time. You have to do a lot of work. So just telling you what you need to know. Don't know what that noise is. Okay, so there you go. You don't have to do this this worksheet. You don't have to send it to me. Right. First question says function groups. What are the functional groups? What are functional groups? Look at the videos I told you in class. Is that basically they're hydrocarbons. That they're molecule groups that can be attached to hydrocarbons. They make very specific types of molecules. The different types that we're covering 2 now. There are several, but we're just going to cover 2 for test 2. And one is a hydroxyl, and that's when you have a carbon with an OH attached to it. I'm leaving the lone pairs off of oxygen. It's called hydroxyl. A carbonyl is where you have a carbon that belongs to your chain, okay, your carbon chain. That has a double bond attached, a double bond oxygen to it. Again, I'm leaving off the lone pairs. For just to get things going here. As far as where are they found, hydroxyls are found in alcohols, and technically they're found in ethers, and carbonyls are found in aldehydes and ketones. So we're going to go over alcohols and ethers, and we went over aldehydes and ketones. So that'll be part of the review. Okay, this first group is alcohols, and alcohols have the OH coming off of it. Okay, and one of the main things you need to know about these guys is know how to name them, and given the name, come up with formula. Okay, given a formula, come up with name. So the idea is that you look at this guy here, and so this has 6 carbons. 1, 2, 3, 4, 5, 6. I'm going to leave the hydrogens off the carbons. I know they're all single bonds. So wherever there's not a bond, there's a hydrogen bond. The hydroxyl comes off of number 1. I could put it at either end. I'll put it over here. So that means I'm going to number it from this direction. And it has a methyl coming off of number 2. And there's 2 of them. So there you go. Okay, 3-ethyl 4-methylheptanol. So there's 7 carbons. 6, 7. Putting in the carbon-carbon bonds, leaving off the carbon-hydrogen bonds. The hydroxyl gum comes off number 2. I'm going to put it up here. And I'm going to go again. I'm going to go right to left. I could have gone the other way. And all right, I'm having a little cough here. And it has a 4 coming off of number 4. 1, 2, 3, 4. So I have carbon coming off here, and it has a ethyl coming off number 3. 1, 2. Whoops. Okay. You have, so it's this number 1 carbon. 1, 2, 3. So the methyl comes off of here, and actually the ethyl comes off of here. I was wrong. I should not be starting from this carbon. Should start at the end. So 1, 2, 3. So this 3rd carbon, 4th carbon. Okay, 3-methyl-3-pentanol. So I draw 5 carbons. 1, 2, 3, 4, 5. Okay, and the pentanol comes off. I mean, the hydroxyl comes off number 3. You could put it off the top or the bottom, and it also has another methyl coming off right here. All right. I also ask you, so that's structured for each one. Also tell you what this should be, what, not hat, what type of alcohol each is each. Right, so we'll figure out the type. You look at the carbon that's attached to the hydroxyl. You count how many carbons are attached to it. This carbon here only has 1 carbon attached to it, so this is a primary alcohol. Okay, so this carbon right here has 2 carbons attached to it. 1, 2. So it's a secondary alcohol. And then finally this guy, this has 3 carbons attached to it. 1, 2, 3 attached to the hydroxyl carbon. Okay, so that's it with alcohols. You have to know how to name them or come up with their formulas. All right, ethers. We won't spend a lot of time with ethers. They don't really show up in the kinds of molecules that we'll be looking at. But just for sake of covering stuff, let's go over them a little bit. So ethers are going to be where an oxygen is trapped between carbons. So what's similarity between alcohols and ethers? They both have single bonded oxygens. So in alcohols, there's the carbon has 2 single bonds to a hydrogen and a carbon. For an ether, the oxygen has single bonds to 2 carbons. All right. So what I'm going to do is give you a name, figure out the formula, or you could go all the way around. So when you have an ether, you have an oxygen, and you have groups attached to it on either side. In this case, you have 2 methyls attached to, one on one side and the other on the other side. Methyl propyl ether, draw your oxygen, and you have a methyl on one side, doesn't matter which side, and you have propyl on the other side. Okay, hopefully that's pretty simple. All right, aldehydes. Okay, aldehydes will have a double bond to oxygen, and they'll always be on the end or the beginning of a molecule. There's 2 guys that kind of go that are oddly named. So if you have a single carbon, okay, it's called formaldehyde, and if you have a double carbon, it's called acetaldehyde. Okay. What makes these differences you'll see in the next example right here. So this guy, let's draw this out. These are special names. Now, if you don't have special name, you have to use the scientific process. So there's six carbons. 7. 7. Okay, since Al tells you it's an aldehyde, so it has a OH at the beginning or the end. I will put it over here. Okay, that means this will be number 1 carbon. This would be 2. I put the bonds in here. Okay, so it's a heptanol, and you don't have to put a 1 or anything in front of it, and it has a methyl coming off of number 2, and off of number 3, and off of number 4. It has the ethyl. Okay. All right. So heptanol tells me the longest chain is 7, and then the add groups. Now let's go back to that formaldehyde. formaldehyde and acetaldehyde. Technically, formaldehyde is also known as methanol. Methanol, because it's a one carbon aldehyde, but we use formaldehyde. And acetaldehyde, it technically is ethanol. But we named them before we knew what they look like, okay? It has two carbons. So if you didn't know their special names, you would call this has two carbons. It's aldehyde, so it'd be ethanol, and this guy would be one carbon methanol. All right. I talked about there's so what you can do is you can take there's a chemical reaction that relates alcohols to aldehydes, and that's if you take a primary alcohol, it can change to aldehyde if it's oxidized. And an example of that is ethanol. Ethanol is primary alcohol. Here's ethanol's chemical formula, structure. Only has one carbon attached to it. Two hydrogens come off. One of the hydrogens come off of the carbon here. Another one comes off here, and so you end up with this. Okay, and this is double bond at the end or the beginning, aldehyde. So you change primaries to aldehyde. Another example of that is one propanol being oxidized. So here's propanol, one propanol. Okay, it has the OH on number one carbon. It's a primary because it only has one carbon attached to it. Oxidized, so lose this hydrogen and another hydrogen off the carbon right there. And so what you get is the OH. And this is the aldehyde because it's at the end. So you can do the same thing with the secondary. Okay, so here's an example of 2-propanol. Okay, so here's that hydroxyl group that's attached to number 2 carbon. Okay, so what's going to happen is two of the hydrogens going to lose. There's a hydrogen here as well. So this guy comes off and this guy comes off, and so what you do, and it comes off as H2, and then what you end up with a ketone. And in this case would be 2-propanone. All right. Notice the double bond is where the single bond was. Tertiary alcohols cannot be oxidized because the carbon, the carbon doesn't have two hydrogens come off. So here's an example of a tertiary. Okay, so here's a tertiary alcohol because this carbon here has 3 carbons attached to it. 1, 2, 3. To be oxidized, you need to lose 2 H's. It can only lose this one H because this carbon doesn't have the other H to be oxidized. All right, now let's go to ketones, and what you're going to do is have to name them or come up with their formulas. So here's a ketone that we have to draw. So ketones are like alcohols. You have to name where the carbonyl comes off. So 1, 2, 3, 4, 5, 6. Okay, my bonds are kind of screwed there. And I want to have the hexanone come off number 4. I'm going to go right to left here, so I'm going to put it right here. 1 carbon, 2 carbon, 3 carbon, 4 carbon. I have 3 carbons coming off. 1 here, actually 2 here, and 1 here. 2 off of number 2. It's a trimethyl, so there are 2 methyls coming off of 2 and 1 off of 3. And there you go. 4-ethyl-3-octanone. 1, 2, 3, 4, 5, 6, 7. 8. Okay, this time I'm going to go right to left. So I'm going to put the double bond oxygen off for number 3. And there's the ethyl coming off of 4 right next to it. So right here. Okay. So for alcohols and ketones, there will be a number in front of the longest chain to tell where to put the OH or the double bonded oxygen. For aldehydes, you won't have to do that. You won't have to put a number in front because it's always going to be at number 1 carbon. And that kind of tells you what the similarities between ketones and aldehydes. They both have double bonded oxygens, okay? The difference is that that's similarity. The difference is that the aldehydes will have the OH at the beginning of the chain, and the ketones will have this double bond not at the beginning or the end. Also, aldehydes are produced from oxidation of primary alcohols, and ketones are produced by oxidation of secondary alcohols. Right. Next thing says show the conversion of glycerol to glyceraldehyde. So here's glycerol. You have to know the structure. We're going to oxidize only the top carbon. Okay. So what's going to happen? This is a primary alcohol because it only has 1 carbon attached to it. This one down here. So that means it's going to lose this H and this H. The double bond is going to go and replace this here, and so you get this right here, or this right here. Okay, so what we formed is something called glyceraldehyde. Right, and I told you is that this type of molecule that has more than 1 hydroxyl and a double bonded oxygen, aldehyde or ketone, is known as a carbohydrate. So we say that they're a many or polyhydroxyl. Polyhydroxyl. And maybe aldehyde or ketone. Okay, so we're ready to start talking about carbohydrates because what we just learned is we learned about alcohols and we learned about carbonyl groups, and when you add them together, you got things called carbohydrates. Okay, so I'm not going to go over this. You can look it up. I mean, you can look in the video. Characteristics of carbohydrates. You know, is one thing is that the monosaccharides will always have this kind of formula. Okay, where N is the number of carbons. So if I know the number of carbons, I know the number of hydrogens, which is twice its amount, and oxygen's the same amount as carbons. So if I have 6 carbons, I will have 12 hydrogens and 6 oxygens. So if you look at the ratio is for every 1 carbon there's an H2O, and that's actually where it gets its name from, carbohydrates. And it looks like it has a carbon with a water attached to it. So we say the reduced chemical formula for all carbohydrates is simply CH2O. And that's where they originally got the name. It has nothing to do with the carbohydrates actually having water on them. And there's a couple other things like they're all polyhydroxyl aldehydes or ketones. A chirocarbon, okay, I go over that, and that's a carbon that a carbon that has one way is it determines how a carbon. It's getting kind of tongue-tied here. A chirocarbon is that if you have 2 different structures and they both have and they look exactly the same and they have a chirocarbon, those 2 structures, even though they look the same, they could be mirror images of each other. because of that chiral carbon. So a chiral carbon in a structure can have a molecule be mirror image of each other. Okay, so you can have so it's like a left hand and a right hand, even though they look identical. Okay, they're different because of how of this carbon, how you attach things to it. And then I told you how you can recognize it is as if chiral carbon has all different things attached to it. It's chiral, and if it has doesn't have that, it's achiral. If it's achiral carbon, that means that structure does not have a mirror image, and the structure that you draw is however you draw it. So if it has a chiral carbon, you have to be very specific how you draw it, and we'll see that when we talk about D and L's, carbohydrates. For instance, okay. So what we did, so what you want to know is, so this is glyceraldehyde. Okay, it's from the previous slides a couple weeks ago. Okay, now what we want to do, and I go over this, is that look at each one of these carbons and decide if these carbons are chiral. And I go over it. So we look at this carbon down here, and I have it condensed, but when we drew it, it looked like like this right here. So this right here is this over here. And the way to tell if it's a chiral carbon is if it has 4 different things attached to it, then it's chiral. Well, you can see it doesn't have 4 different things. It only has 3 different things. So this guy is not a chiral carbon. Right. Now the second carbon up is this chiral. Well, okay, look at it. It had this. What does it have? It has an H attached to it. It has an OH attached to it. Is this thing attached? Don't look at the carbon. The whole thing. It has this attached and this attached. So yes, this is a chiral carbon. And I'm going to circle it. Okay. Now how about this carbon? This carbon has well, how many things does it have? Well, it only has 3 things attached, so it has to be 4 different. So this is what we call achiral carbon as well. So this guy, it's important how we put this OH. If we put the OH on the right or left, it's going to actually be 2 different structures. And we're going to see for all our carbohydrates, the second carbon up, if it's D, the OH is going to be to the right. If it's L, it's to the left. For just about all of the ones that we're going to talk about are always going to be D, so the second carbon is going to always have, if it's a D, going OH. So OH to the right is D, OH going to the left is L. Okay, so we're going to go into the structures of the other carbohydrates. One thing you'll notice in the other structures of the monosaccharides, they all will have this in common, if they're Ds. All of them except for fructose would look like this. Okay, we'll have this at the top. It's the middle ones that will be different. So you're going to have to pay attention to that video I gave you on. I'll quickly go over it in this PowerPoint. Okay, so we're going over these 2 monosaccharides. Okay, ribose and deoxyribose. These are called pentoses, and they're called pentoses. Ose is the magic ending for sugars, like glucose, cellulose, and so forth. Fructose. So this tells you have a sugar. Whenever you're ose, you have a sugar. So it tells you have 5 carbons. So draw out the 5 carbons. 1, 2, 3, 4, 5. All right, draw those out. Put this at the end, okay, and this at the top, just like for glyceraldehyde. Now, in the middle carbons, you got to decide where to put. So in the middle carbons have OHs coming off of them. Where do they go? So it's very specific where that you put them, right or left. Okay, kind of way I remember is RI, right. RI, right starts with I, and so is ribose, so both of those go to the right. And there you go, there's your structure. Okay, deoxyribose is just like ribose, but the second carbon is missing an O. All right, right there. That's why they call it deoxy. So if you draw this one first, you should be able to come up with this one. Okay, how about for the 3 hexoses? Okay, so you should know how to draw the 3 hexos. Hexos means 6. Ose means it's sugar. There's 3 of them: glucose, lactose, and fructose. We're going to cover these 2 first. So let's do glucose first. So it has 6 carbons: 2, 4, 5, 6. Bottom 2 like that. Top 1 like that. Let's do the same thing for galactose. 1, 2, 3, 4, 5, 6. Draw that and that. Okay, now let's do. So now there's 3 middle carbons. How do you put the OHs? Okay, well, for glucose, they go right, left, right. You might want to think of run, little rabbit. Okay, right, left, right. And for galactose, it goes run, right, left, left. And so the bottom one is going to be different. So you can call run, little leopard or lizard or whatever. Okay, so that's how you do these. So if you get this guy, you can figure out this one, all right, or vice versa. Okay, so tops and the bottoms the same for so far for all the ones we talked about. Okay, now let's do fructose. Okay, fructose is going to be a little bit different. Okay, so fructose is a little bit different. So what you do is you draw. The way that I do it is I start out with glucose. I draw out glucose 1, 2, 3, 4, 5, 6. Draw this guy, then draw this guy, and then it's right, left, right. Okay, and then what you do is you swap these 2 guys. So the OH goes up here, and notice now is OH is up here, and this O is here. So it still goes, so if you look at it, it still goes right, left, right. Okay, so top, now the top is different. So the way I like to do glucose is, fructose is draw glucose, then swap the tops. All right, on the way that you find these monosaccharides in nature is actually not the linear form, it's the cycle form. And this is glucose, and we can count them. Each point is a carbon, so 1. And we go clockwise, so this is carbon number 1. You always start over here at this end. So carbon 1, 2, 3, 4, 5, 6. This carbon right here. This is not 6 right here, all right? So you're counting the carbons. Carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. Okay, the second thing you need to know about this structure is this OH, only this OH. Okay, if it's pointing up, it's called beta. If this OH was pointing down, like right here, then it would be alpha. Okay, so you have to know how to number each carbon in a ring structure and tell me if it's alpha or beta. Okay, the next thing, so that's the structures of our monosaccharides, and we went over glucose. Now what you want to do know is their function. All right, so I go over this, and I spend a lot of time talking about this in the PowerPoints. And I'm just going to briefly go over this. So glucose is found in all living things, and that's because it's used as a primary energy source. All our cells need glucose. That's primary energy source. I show you how it's made. It's made from photosynthesis, and I go through that chemical reaction and how it turns into, and how plants take what we produce from the combustion of glucose to make its own glucose. Then I talk about how it moves around the body, how it moves from your gut to your cells, and then I also show how it's the cells, how it's broken down in your cells. Okay, and this is called cellular respiration. And there were 3 steps. There was glycolysis, which breaks the glucose in half. There's a Krebs cycle, which breaks off the carbons and the oxygens. Okay, and then the hydrogens are passed on, and oxidative phosphorylation, and these combine with the oxygen you breathe to form water and energy to make ATPs. And the ATPs is what makes you do your work. Now I also talk about how it relates to diabetes and low blood sugar and so forth. Okay, this slide is just a repeat, and let's skip it. All right. So I'm not going to go over these; they're already in the video. So you need to know what function fructose and what function galactose has, and you can get that from the videos. All right. So that deals with all the polysaccharides. Let's see how many were there. There were the glyceraldehydes, and there were 2 of them, the D and L. There were the 2 riboses. Okay, D ribose and D deoxyribose. And then there were the 3, let's say there are 2 glycerides, 2 ribosomes, and 3, this is pentoses, 2 pentoses. And then there's 3 hexoses. Okay, there's glucose, there's galactose, and fructose, and these are all the D versions. So, I have to know how to draw these, or at least how to recognize how they're drawn. All right, next thing is disaccharides. Disaccharides are where you have 2 of these monosaccharides linked together, and we're just going to talk about 3 of them here. There's maltase. Okay, there's maltose. We'll get to maltase in a second. There's lactose. And there's sucrose. Okay. Right, maltose is made out of 2 glucoses. Right, and the bond that holds those together, all these guys together, are called glycosidic bonds. Now, all 3 of them have 3 different types of glycosidic bonds. Okay, so if you notice, you have a glucose here and a glucose here, and the bond here is connected to the 4th carbon. So 1, 2, 3, 4. And it's attached to the 1st carbon over here, so it's 1, 4. Since it's pointing down, just like when we were talking about the alpha beta, down was alpha. Since this whole thing is, so this bond is pointing down, alpha 1, 4 glycosidic. For lactose, you have a galactose and a glucose, and it has a glycosidic bond. The bond is pointing up, so it's beta, but it's between the same carbons, 1 and 4. Okay, sucrose has a glucose and a fructose, and they're attached top to bottom in a sense, where there's bond pointing down and a bond pointing up. and they're connected to 1 carbon and the 2 carbon. So it's a glycosidic bond. So it's going to be between the 1 carbon and the 2 carbon. So we call it 1, 2, going down, going up. So 1, 2 alpha beta glycosidic bond is found in the glucose. So notice you have to recognize, so you have maltose will look like that, where you have the glucose pointing down, so it'd be alpha 1, 4. Lactose, okay, you have lactose has galactose and glucose, and it's pointing up, so it's beta 1, 4. And then you have sucrose, or glucose, fructose, and that's 1, 2 alpha beta glycosidic. Right, so you have to recognize this types of bonds. Now, when we eat these guys, when we eat maltose, our bodies break down the maltose into the 2 glucoses, and the way it does that, there's an enzyme that will break that bond right there, and then it'll release the glucose so that we can use it in cellular respiration. When we eat lactose, there's an enzyme called lactase which will break these 2 guys, so the galactose and the glucose will go off. And then finally, sucrose. When you eat sucrose, there's an enzyme called sucrase which breaks that bond to release glucose and fructose. And I go over the function of each, okay, and in the PowerPoints. Right, and finally there's polysaccharides, and the polysaccharides are many monosaccharides all linked together. Okay. Okay, and they can be really big and really long. And so this is really the macromolecule of carbohydrates. So the long chain made out of monosaccharides. We're studying 3 of them. We have starch, we have glycogen, and we have cellulose. What's common between them all is they all made out of glucose, are the monosaccharide that makes them up. Okay, so starch. Okay, starch is made out of 2 types of polysaccharides, amylopectin and amylose, and I go over that. And the video. One is more efficient for burning quickly. One is more efficient for burning long term. Like I told you, like Kindle. One is like Kindle. One is like giant logs. I go over where is it found. It's primarily found in the seeds of plants, and the reason why it's to produce the energy so that the seeds can grow. And you can see that. And why is it good for us is because we can eat starch, and when we eat starch, it's made out of many of these glucoses, okay? And when we eat the starch, our bodies will break the glucoses off, these guys, and these glucoses can travel into our bloodstream and get to our cells and give us energy. So that's why plants are really good for us, because they can give us this give us this source of energy. All right, glycogen. Okay, that's plants use starch as a way to store glucose. We use glycogen, and I told you how the structure looks like. It kind of looks like amylopectin, I think, or amylose. Where's it found? It's found primarily in our livers and our muscles. It also found in other cells. And then I go over why these are 2 really good places to store it. Why is it stored in your liver, and why is it stored in the muscles? Okay, the liver. This is a good way to replenish blood sugar, and this is a good way to preserve blood sugar, because muscles has its own source of glucose. But please listen to the uh, video, and I also talk about how it moves all the way through your body, and etc. This is the big guy. This one's really, this is the one you should know about going into your fields. Okay, cellulose. Okay, cellulose. Again, is a bunch of glucoses all stuck together. Okay, but they have special kinds of bonds, and so uh, that are different than glycogen and starch. And I'll get to that in a second. But these are found in plants, and they're usually like these stems or like tree bark and things that, if you were to eat a plant, this is the part that's you can't eat. Okay, in a sense, it's kind of like the fiber. So when they talk about fiber, if you eat, some vegetable, some of it you're not going to digest, and it's because it's made of cellulose. And why is that? Okay, so technically, cellulose is made out of a lot of glucose. So technically, cellulose is a huge energy source for glucoses. Okay, the problem is that if we were to eat, and when we do eat cellulose and like fiber, our body can't break that bond, and therefore these glucoses just stay on this chain, and they cannot be metabolized. It's a shame. It's unfortunate to us, because if some reason we could have the ability to break the glucoses off these chains, we'd have a huge energy source, okay, food source. You could go around, every time you eat a plant, you can get everything you need out of it. I mean, you can eat the whole plant, and the plant would digest. Or you can go up to a tree and start gnawing on a tree. Or you could take your homework and eat your homework, okay? They talk about, my dog ate my homework. Well, you could eat your homework, because the paper that you eat is made out of cellulose. And if you had the ability to eat this, when you ate it, you could break off the glucose, and then you could get energy from it. Unfortunately, we can't do that, but there's some animals that can, like termites. Okay, so that covers the review. Obviously, you're still going to have to look at the videos and get specific things, like what are the functions of glucose and glycogen and blood sugar, chirality, and so forth. Okay, so there you go.